Hello everyone, welcome back to Biotechnica channel. We are going to have a series of important question discussions from previous year question papers from CSIR Life Science NET exam. And today's topic is from Unit 1 and specifically questions from Nucleic Acid Structure. As we know in CSIR, nucleic acid is a very important topic and there are quite so many questions from this area which are based on the structure, the topology, the different properties and also some of the stability questions. Let's look at all of this. We are not keeping the metabolism question today because that is coming in a separate subunit. Maybe some other time we will discuss with other metabolism questions. There was a question in September 2022 regarding the stability of DNA. It represents the figure denaturation and renaturation profile of a double-stranded DNA in citrate buffer. What is the percent of DNA that remains denatured at 30 degrees centigrade after it is cooled down from 100 degrees centigrade? Here are the options and here is the profile, the graph which shows the denaturation and renaturation of a dsDNA. Now in such kind of questions, you must always, first of all, if it is a graphical question, you must always see what is the y-axis and what is the x-axis. Here, as the temperature increases from 30 degree to 100 degree, you can see there is an increase in absorbance. Second, for such type of questions, you should see whether it is the denaturation or renaturation, double-stranded, single-stranded. These properties, whether it is DNA, RNA, these properties you must keep in mind. Apart from that, some characteristics of DNA is that it is having the property where if it is denatured, then the two strands, they are separated. As we know in a DNA, the strands are staying together, but if it is denatured means the strands will separate, which is also called as melting. Now what happens when the strands are separated? The bases, they are exposed. What are these bases? These are nitrogenous bases which are aromatic in nature, and being aromatic, they will be taking absorbance. They will be absorbing some wavelength of light at 260 nanometer, and this particular absorbance, when it is in the double-stranded DNA, the absorbance is less. How much it will absorb wavelength of light, but when it is in the denatured form, the absorbance will sharply increase. That is how we come to know that the denaturation has taken place. Here in this example, in this particular question, if you see what is the absorbance of the double-stranded DNA initially, when you start this graph, you can see that the absorbance is just 1 over here. Okay, but when you have started with the denaturation, at some point of time, you can see that the absorbance is increasing. After a certain point of heating, the absorbance is continuously increasing and it reaches the absorbance of 1.40. Okay. Now from here, we are seeing that it is cooled down. It is written that at 30 degrees centigrade, how much of the DNA will still be denatured after it is cooled down from 100 degree. So again, it is cooled down from 100 degree. You should follow what is written and you should match with the profile what is exactly happening over here. So when it comes down, you can see that when the cooling is done, you can see that absorbance, it is coming to around 1.12. So this is a trap question, you can say, where students might make a mistake that if it is renatured, then it will again come back to 1. That is not the case. It comes to 1.12 as we can see. The absorbance is 1.12. It means that some of the DNA, it is still in the denatured form. That is why what we have to do is we have to subtract from 1.12, we have to subtract 1. Because initially before denaturation, entire of DNA means 100% of DNA, it was in double-stranded form. And the absorbance was 1 at that time. But now when entire DNA was denatured, the absorbance is just 1.40. So following by this renaturation process, was pushed and some of the DNA still remained in denatured form. That is why we will get an absorbance of 1.12 in place of 1. Okay, so 0.12, 1.12 minus 1, 0.12 is the actual absorbance of the DNA, part of which did not renature at all. So they are still in the denatured form. So you have to find what is the percentage of this. Okay, so what you can do is, you can, if you know at 1.40, all the DNA was denatured, so we can say at 1.40 we have 100%. And if we take unitary method, then it will be 100 by 1.40, and this is the part which did not renature. So 100 divided by 1.4 into 0.12. That's why the answer will come to 8.57, and going by this, option 1 is the right answer. Okay, the next question is also from CSIR, question September 2022. Here it is regarding the diffraction patterns of two forms of DNA, which is A and B, and the statements which are related to these patterns, they are given below. Now here, this is form 1, which shows a typical X structure. Many of you would have seen this particular diagram in the books, in standard reference books while studying for nucleic acids. But however, we will not pay attention that whether it is A DNA or B DNA, what is mentioned. Okay, so these are some small hidden things that we take for granted and we tend to ignore while during our preparation. But CSIR targets those parts. Fine. And they have given another such diffraction pattern, which is from form 2. Okay, so of late historical questions also they have started giving, like experimental questions which are already done in the past. Okay, now the diffraction pattern corresponding to A DNA, it is shown by form 1, which is 11.6 base pairs per turn. Either this or the diffraction pattern which is shown in form 1, it is B. Okay, so B DNA shown in form 1 with 10 base pairs per turn. Similarly, here also they have said form 2, it is either for B DNA or it is for A DNA, and these many base pairs per turn will be there, isn't it? So all you need to know, anyways, people will be knowing whether what contains how many turns. Now we all know that the Watson-Crick pattern, which was the B DNA, so it contains around 10.4 base pairs per turn. However, we can just roughly write that it has 10 base pairs per turn, right? So anyways, we know for B DNA it has to be 10. That means D should be the correct answer. Okay. So in fact, this is the form 1 is what is actually for B DNA, and that has 10 base pairs per turn. So like this you can use, and then obviously form 2 is a different pattern. It is not the same pattern. So that's why this has to be A DNA. Okay. So the diffraction pattern of A DNA, it is shown in form 2. Not this one, but it is in form 2. And that has 11.6 base pairs per turn. So that is why C and D is the right answer over here. Okay. So we know that Rosalind Franklin and Raymond Gosling, they had discovered this X-ray diffraction pattern, which was produced by a DNA fiber. And according to this experiment, the historical X-ray fiber diffraction patterns, they are A DNA and B DNA, which are plotted on the same scale. After completing the qualitative description of the A DNA pattern in comparison to B DNA, it was found that the A DNA, the number of nucleotides is more per helical period. It is closer to 11 per strand instead of 10 in B DNA. Okay. So here are the diffraction patterns as we just saw. The X-ray, the typical X-ray, actually these are the different bases which is seen from the above, that is the helical axis. And the base pairs per turn is 11 because A DNA is a more shorter DNA. Okay, it is a more compressed DNA. This happens when the humidity is less, and that is where kind of A DNA shrinks. So as you can see, the A DNA is a shorter one, and B DNA is the regular Watson-Crick pattern, right? So this pattern, what you see in this A form, the 8th layer of the line or layer line of pattern, it occurs at about the 10th layer line of pattern D, right? So this reflects a 20% increase in the DNA helix period. Now the A DNA pattern shows crystalline spots, right, on the first few layer lines, whereas the B DNA pattern, it shows broad streaks, right? Then A has immediately apparent significant difference between the two patterns is the presence near the center of an X shape. It is a 3-layer line in B, and that 3-layer line it is missing in A. So this X shape is produced by the regularly repeated part of the molecule, which is nothing but the sugar phosphate backbone, and the X shape clearly appears in pattern B, which reflects the scattering by an individual angularly averaged molecule, whereas it cannot be discerned in pattern A because the pattern is resolved in individual crystalline spots near the center. So as you can see in aDNA, the bases are more pushed towards the corner, means periphery, and in the center they will be missing. Okay. And also B shows a diamond pattern, which not only means helix, it also reveals the 3 fundamental dimensions of the phosphate helix. Okay, that is the pitch from the layered line interval, then radius from the meridian angle of the X shape, and also the nucleotide repeat from the vertical diagonal of the meridian diamonds. Okay. So we also have one more question regarding this from December 2019, which is again regarding A and Z DNA. That is different forms of the DNA, right? So A and Z forms, they are not so common, but they will appear sometimes. They are the double-stranded DNA, which differ in the handedness of their helices, nucleotides, and configuration of the base to sugar. Now, based on these properties, which one of the following defines a correct combination of A and Z forms of DNA, right? Now, here we have either right-handed double helix and anti-conformation for the base to sugar arrangement in A, that is in aDNA, whereas left-handed double helix with alternating sequence of G and C as a general pattern and alternating syn and anti-configurations for the base to sugar arrangement in zDNA, right? Now, if I show you these two concepts, if you are not aware of, these are the glycosidic bond conformations which we observe in nucleic acids. We have this ribose sugar and along with that the nitrogenous base. So, if our the carbon, the 8th carbon of the purine nitrogenous base, if it is near to this carbon 1 dashed of the ribose sugar, hydrogen is attached with the carbon, that's why it's written H, then it is called as anti-conformation. Okay, so it's just that this bond can rotate. If this bond can rotate, this entire base will be rotated this side. In that case, this carbon 8 will be facing this side of this carbon 1 dashed. So if this is the case, then this is called as syn conformation. Okay, so we have two conformations, one is syn, another one is anti. And what we have in aDNA, in fact in bDNA also we have anti-conformation of bases. But in zDNA we see alternating anti and syn, because in zDNA we have this this alternating conformations, that is GC, 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 like that we have. Okay, so sugar is in zDNA, first of all it is left-handed. Okay, and why it is called as zDNA is because generally our backbone of the DNA that will be straight only, means it will not be having any zigzag. But in zDNA the backbone itself is zigzag. Okay, it is a helix only, but the backbone is zigzag like this. So this is what is a zDNA. Why the backbone is zigzag? Because generally it will be seen in stretches of this GC-GC alternating bonds. Okay, and when this is the case, our C, cytosine, is in anti-conformation and guanine is in syn conformation. There is also the sugar puckering, which is 2-dashed endo and 3-dashed endo. What is that? In this 2-dashed carbon, in 2-dashed endo, it is slightly above the plane. As you can see, this is the plane of the sugar, this is slightly above the plane. Whereas the G in 3-dashed endo, the 3-dashed is above the plane, but 2-dashed is below the plane. Okay, so we will call this as 3-dashed endo, or we can also call it as 2-dashed exo. Both are the same thing. Okay, and here also we can call this as 2-dashed endo, but at the same time we can call this as 3-dashed exo as well. So this is what we have. Our guanine is in this 3' endo and base is in syn conformation. Because of this alternating conformations, when the bases, they will pair up. So if we roughly take the shape like this, and if we roughly take the shape as like this, okay, so one base is like this and another base is like this, okay, then again one base is like this, then another base is like this. Now from the other side also, if we take, so one base is like this, that is the G, then the next one will be C, so it will go like this, okay, then again base is G, then again it is C, it will go like this. So you can see here is the base pair, here is the base pair, here is the base pair, and here is the base pair. You can see a typical Z form. That is why we have the Z DNA. Okay. So this explains why option 1 is right over here. It's a right-handed double-handed helix. Okay. Then we, so just wanted to make sure that you are aware of this concept table book. You can see here, this is one of Biotechnica's revision tools, where A, B and Z DNA, the different classifications are given. Actually, it is a book where such tables are made, comparison tables with all the details. As you can see, just now we saw A DNA is anti-conformation, B DNA is anti. So this is a very good resource for revision once you have prepared your topics. Okay. Then I just said we have alternating syn and anti and endo and exo. All this we have, right? Then we have another question from CSIR November 2020 exam. This is also regarding the base pairing. And here they are asking this particular sequence, which out of this can be possible and which is incorrect statement of here from here. Okay. Now you might think that, okay, U is there in the sequence, which means DNA cannot be there. Right? But actually, if you see here, obviously DNA can form. Uracil in DNA, it is not a regular base, but it can result from deamination of the cytosine to uracil, which can create a mispair. Okay, so you can have this AU, you can have UG mispair, or you can have it from incorporation of DUMP instead of DTMP. So that will lead to a UA pair. Like this, you can have a base pairing even in DNA also. It is not restricted only to RNA. Okay. So that again, it's kind of a trap question. People might hurry up in coming to the conclusion of answer. But actual answer over here is option 4. Why? Because the RNA molecule cannot be in B form. If anyone knows this, the RNA molecule cannot be in B form. So here, if you have noticed in B DNA and A DNA, in B DNA the sugar puckering is 2' endo. Okay, which means if I show you here, you can see the 2' endo, it is pushed up above the plane. So this is what we have in B DNA, right? Now one property of RNA, what is that? It is ribonucleic acid, isn't it? So in RNA, what do we have? In DNA we have deoxy, but in RNA the oxygen is present, isn't it? So where is the oxygen present in RNA? It is over here. That is OH, this O will be present, isn't it? Now, if the 2' endo is pushed upwards, and here itself I will have the base, isn't it? So here I will be having the base molecule. So this oxygen can create a steric hindrance. That is why RNA, if it is a RNA sugar, then it cannot be in the B conformation. That is not possible. Yeah, if it is DNA, then here it is only just hydrogen, which is only one small atom. That is why even if hydrogen is present, the base will not have steric hindrance. So while DNA can be in B form, but means RNA can never be in B form. This is actually a very in-depth concept which you will require. Okay, so it cannot be a double-stranded RNA molecule, and that too in B form. It probably could have formed a double-stranded RNA, but then it can only form in A form helix, where the 3' endo will be pushed upwards. So 2' if it is down, then it will not create any hindrance with the base. Okay. Let me know in comment section whether you learned anything new here. whether you were not aware of this, and if you have learned today, then let me know in the comment section, and you can also let me know which property you have learned new today. Okay. Then another question very recently, it is from December 2023. That is a recent exam that we had. So which of the following nucleic acids with same concentrations in water will form a stable stem loop structure upon annealing by heating and flash cooling on ice? Okay, so we have here our several sequences. What are they asking? That when it will form stem loop structure. Now stem loop means what? It will have a stem like this, a loop like this. Now why stem will form? Obviously by base pairing. Why loop will form? Here the bases don't pair up with each other. So here this side bases and this side bases, they will not pair up. But in this stem structure, the bases, they will be pairing up. This is the concept that you need to know. That means first of all, we know RNA. This is our mRNA, our rRNA, and our tRNA. So in these you can see stem loop. It is nothing but intramolecular base pairing, right? So it is a pattern that occurs in single-stranded DNA, or more commonly it can also find in RNA also. The structure is known as hairpin or hairpin loop, where you can see some pairing is there, base pairing is there. You can have some bases which will not pair up. It's not necessary that all will pair up together. Okay. So it occurs when, but maximum pairing should be there. It occurs when two regions of the same molecule, they have palindromic sequence. And the first prerequisite is the presence of a sequence that can fold back on itself to form a paired double helix. So what you should look to is whether from the 5-10, from the 3-10, the bases are matching or not. So we must check the base pairs from both the ends of each sequence. Okay. And while doing that, we should keep in mind that pairing between guanine and cytosine, they have 3 hydrogen bonds, and they will be more stable compared to adenine uracil. So it should be noted that in RNA, guanine uracil pairing, it features only 2 hydrogen bonds. Okay, they are common, and in those cases they will be favorable. Okay, so guanine uracil, they will have 2 and they will be more favorable. So from here, if we look in the first sequence, the G in the first and the last base, which is also G, they cannot pair up. Okay, then we can see G in the second base, G in the second last base, they also cannot pair up, CC cannot pair up. So 1 cannot be the answer, right? Same with fourth sequence also. If you see A and G, they will not pair up because both are purines. Okay, then G and G will not pair up. A and C. So like that, bases from 5' and 3' do not pair up to form stem loop structure. Now in second sequence, if you see, the first base being C and the second base being G, so they will pair up. Okay, and so will be the rest of bases in this sequence, because C and G will pair up, then G and C will pair up. So there are more bases in this sequence which will pair up like this, and that is why this will have the more chance of forming a stable stem loop structure. In third sequence, first base A, and the last base A, U will pair up. Okay, but then second base, if you see, U and U will not pair up. Then G and C will pair up. Okay, but then again, then C and G will pair up. Okay, but if you see here, more chances are there that in second sequence they will pair up more. So in this sequence, only few bases are there which will pair up. So that is why the best possible answer is 2 over here. Okay. So that is all for this discussion. You can let me know in the comment section which topic you want me to discuss, the questions for which topic you find it hard and you want me to discuss important previous year question papers, questions from Unit 1. Thank you everyone for watching.